Welcome to Chapter 25, Part 1, Microbial Diseases of the Digestive System. This chapter fulfills objective number 4. In Part 1, we're going to talk about the structure and function of the digestive tract, periodontal disease, mumps, Helicobacter peptic ulcer disease, staphylococcal food poisoning, and hepatitis. In Part 2, we're going to talk about shigellosis, hemorrhagic E. coli, Clostridium difficile associated diarrhea, cholera, viral gastroenteritis, giardia, cryptosporidia, tapeworm, and trichonellosis. The function of the digestive tract is to ingest, digest, and absorb nutrients. What's left over gets eliminated. So let's discuss the structure. The basic structure of the digestive tract is a long tube. You start out in the mouth, goes down the esophagus, part of which, the pharynx, we share with the respiratory tract, go into the stomach, then we hit the small and then the large intestines, and then whatever is not digested goes out the anus. Now we also have associated organs. You have the teeth, which are part of enabling us to ingest and digest food. You have the tongue. You also have the salivary glands that produce saliva. You also have the liver, which produces a lot of enzymes which are concentrated in the gallbladder. The digestive tract, as far as your microbiota goes, is also home to the greatest number of bacteria and the greatest number of species, most of which are in your small and large intestines. Not very many bacteria make the stomach their home because of the low pH. You do have a fair amount in your oral cavity. Not as much as in your intestines, but what your mouth and your intestines have in common is it's moist and it's warm and food is going by all the time. With most of these bacteria, we've worked out a good mutualistic symbiosis. We take care of them, they take care of us, they help us out. In fact, recently we've found that the microbiota in your intestines are influential in digestion, in stimulating your immune system to attack bacteria that aren't our friends, and also help in regulating our immune system so it doesn't go too crazy. In fact, they're so important that we went to the effort of figuring out the genetics of all of the microbiota within the gut so that we can understand our friends. And when you take antibiotics that may decrease your normal microbiota of the gut, how we can put it back. In the rest of the chapter, we're going to talk about diseases of the digestive tract, and these usually arise in two ways. One, a strict pathogen comes through and causes disease, or two, the normal microbiota gets disrupted in some fashion causing disease. Those are the two basic things that we see. So, let's take a look at them. Let's begin in the oral cavity. Your mouth is home to an awful lot of bacteria, as I mentioned before, and there are certain diseases associated with the oral cavity. The only one I'm going to cover is periodontal disease. Let's take a look at the structure of the teeth and gums and look at what periodontal disease is. Okay, so your teeth are hard surfaces, the only surfaces within your digestive tract that are not continually shedding cells. So they make a lovely place for bacteria to grow. They're embedded into your jaw, and your gums or your gingiva come up and cover the roots. Okay? And you have your hard enamel around the outside. Now, healthy gingiva or healthy gums come right up to the bottom of the crown area, and you do have spaces between the teeth. In periodontal disease, you get bacteria that grow in these spaces and cause inflammation of the gingiva. Starts out as gingivitis. As this inflammation increases, the gingiva pulls back, you start getting damage to your bone, and you end up with periodontal disease. If it goes on long enough, and the roots of the teeth start to become exposed, the teeth either have to be extracted or they fall out on their own. Okay, so that is the end result of periodontal disease, is loss of the teeth, and you have to wear dentures or get along without teeth. Current thinking on periodontal disease is that it's a disruption of healthy microbiota, that you get some organisms that become dominant that lead to disease, whereas if we had other organisms that are dominant, then you have health. This type of disease does not follow Koch's postulates, so it's very difficult to figure out if that's exactly what's going on. But one thing that we do know is that a young biofilm is better for health than an old biofilm. In other words, if you have, growing in this space, a biofilm that has only been there for a couple of days, it's more likely to lead to health than a biofilm that's been there, well, we'll say a week or more. As biofilms age, the number and types of bacteria change, and we think that's what's leading to this state of non-health, and your immune system recognizes these different microbes, starts to fight the disease. The biofilm is producing products that are damaging the tissue, and you end up with gingivitis, and if it goes long enough, ends up with periodontal disease. If you physically disrupt the biofilm at least once a day or every 2 days, you're less likely to get gingivitis, which is less likely to progress to periodontal disease. So what do I mean by disrupting it? Brush your teeth, floss. Okay, those two things we've found prevent gingivitis. You're disrupting those biofilms, they have to start all over again, and it's a young biofilm. Once you have periodontal disease, we don't know how to cure it. We can keep it under control so you don't lose your teeth, but we don't know how to cure it. So prevention is the best thing in this case. Brush and floss your teeth. One of the organs associated with the oral cavity are the saliva glands. Now, saliva glands, as you can guess from their names, produce saliva. There's a set right in front of the ears by the jaw. Now, mumps is an infection of these saliva glands. So you can see this poor guy is swelling on one side. What happens is you breathe in the virus, so it's respiratory transmission, and it replicates within the lymph nodes of the neck. Okay, right down here. Then from there, it moves up into the parotid glands here and cause swelling. That's what we usually think of as mumps, is that chipmunk look. You get fever. You also get a viremia. You can pull virus from the blood. Now, mumps is painful at this stage. Usually kids and adults that get mumps don't want to eat because, especially with sour things, that causes those swollen saliva glands to try to release saliva, and it's quite painful. But if it were just that, it wouldn't be a problem. The problem comes from the viremic stage. It has a tendency to replicate within the testes. This happens in approximately about 20 to 50% of men. And in rare cases, this can cause sterility. So you've heard that if you get mumps as a teenager, if you're a guy, can result in sterility. It's a rare side effect, but why risk it? We do have a good vaccine for mumps. It's a live, attenuated virus, and we include it with measles and rubella. Now let's continue down the digestive tract into the stomach. Now, the structure and function of the stomach is a big muscular sac, basically, that its job is to mix up the food that's been ingested and expose it to acid, which helps break it down. There's also some enzymes that are produced that help break down proteins, and those enzymes function best in a low pH. Now, to keep the stomach from being digested by its own hydrochloric acid, it has a lining of mucus that protects it. Ulcers. Ulcers are sores that develop in the lining of the stomach. Okay, that's an ulcer. And it can progress from there to bleeding ulcers where they get far enough down that they hit the capillaries and you start to bleed into your stomach. Now, there's also inflammation associated with it. With chronic inflammation anywhere in your body, there's damage to surrounding tissues because the neutrophils are producing nets and they're releasing hazardous toxic chemicals. And because there's damage to cells, cells that are surviving can have damage to their DNA that can lead to cancer. One rare prognosis for peptic ulcer disease is that you will develop stomach cancer, gastric cancer, and your stomach has to be removed, and you have to go on a soft food or liquid diet for the rest of your life. It used to be thought that stress caused ulcers. That when you have stress, you produce more acid. It was thought that the acid would reduce the mucus layer and cause areas where the acid could get to your stomach and start to eat away at it. In Australia, there was a doctor that worked with peptic ulcer disease and a pathologist that looked at the removed cancerous stomachs, and they noticed that in most of the cases you had this spiral bacterium. Okay? And they wanted to get funding to find out if this is what was actually causing ulcers. Is this infectious agent? Well, it's kind of hard to get funding to do this kind of research when the organism only grows in humans. And Helicobacter pylori, H. pylori, only grows in humans. We do not have an animal model for it. So what the physician decided to do is they were able to grow this organism in culture. He got some and made sure he knew what antibiotic it was sensitive to, and he drank it down. And they followed his symptoms, and he started developing peptic ulcers. When he took the proper antibiotic, it resolved itself. By the way, they've both received the Nobel Prize, if I remember correctly, for this one. And it was found that most ulcers are caused by H. pylori. What H. pylori is doing, it can survive in the high acid, but it prefers a little more neutral environment. So it gets down, attaches to the ciliated cells of your stomach, and they produce what's called a urease enzyme. In other words, it breaks down urea, raises the pH, and creates a more favorable environment for itself. Well, when you do that, it breaks down the mucus layer and exposes the stomach to your acid. So in other words, you still have the ulcer being caused by the acid, and that causes damage to the stomach. Now, when we are checking to see if you have H. pylori or Helicobacter peptic ulcer disease, we can do a number of tests. We can have you drink radioactive urea. The urease breaks that down, and you exhale radioactive carbon. So we can do that to see if you have H. pylori, or you also develop antibodies to H. pylori, so they can do a simple blood test. Number of other tests, but they can give you antibiotics and clear up the ulcer disease, which is wonderful. It's a whole lot better than what we were doing for ulcers before, which was simply lowering or raising the pH of your stomach. Now, an interesting side note, we've found that approximately 95% of the population is positive for H. pylori, but not all of them have ulcers. So we're still working on the epidemiology to find out what causes the actual disease. The H. pylori is implicated, it's part of it, but what causes the balance to tip in the favor of the bacterium causing the disease? Let's move on to staphylococcal food poisoning. This is also what you consider to be the 24-hour flu. Now, you know what staphylococcus looks like. You've been looking at it in lab all semester. But this is a good time to talk about the difference between an infection versus an intoxication. An infection is when you get the microbe into you and it's replicating and causing disease. An intoxication is where you ingest large amounts of toxin and that causes disease. Now, we also think of intoxication as drinking large amounts of alcohol. Believe it or not, in large quantities, ethanol is a toxin. It causes, drunkenness and hangovers, and that's kind of a temporary disease. All right, so staphylococcal food poisoning is an intoxication. We have other diseases where staphylococci replicate within the body and cause disease. But in staphylococcal food poisoning, the 24-hour flu, we're talking about toxins causing disease. Now, there are 4 things that lead to food poisoning by staphylococcus: inoculation, high protein content, mesophilic incubation temperatures, and time. Let's talk about each of them. All of us have staphylococci of some sort or another in our microbiota. Now, the one that causes the problem is Staph aureus. It's an opportunistic pathogen. They figure between 20 to 25% of us are carriers for Staph aureus. Doesn't cause disease. But if we get Staph aureus, onto our hands, we can inoculate it into our food. Now, if it's a high protein food, Staph aureus says, woohoo, time to party. So things like dairy products, meat products have a tendency be more associated with staphylococcal food poisoning than, say, lettuce or celery. Staph aureus just likes that high protein content. Speaking of high protein content, most of the time when we cure meats with salt, that takes care of most bacteria. But is it going to take care of Staph aureus? No, because it's halotolerant. It's a skin bacteria. It's also a mesophil. It likes room temperature up to body temperature. So that's where it's going to replicate the fastest, are in mesophilic temperatures. They can also handle fairly high temperatures, clear up to 40. There are some mesophils that are sensitive to those higher temperatures. Staph aureus is not. And then time. So here's a scenario. Be ready to be grossed out. Okay, so I'm a food service worker and I'm making chicken salad. And I pick my nose and don't even think about it and proceed to finish making my chicken salad. Now, good aseptic technique. Do not touch your face while you're doing food. If you never do that, then you don't have to worry about inoculation. So my Staph aureus has been inoculated into the chicken salad. We have high quantities of egg. We have high quantities of protein from the chicken. And if we put it in the refrigerator immediately, that Staph aureus is going to be inhibited in its growth because it's below its optimum temperature, and it's not going to replicate quickly. But let's say we leave the chicken salad sitting out at room temperature for a couple of hours. They're going to go crazy replicating within that chicken salad, and they're going to start producing toxins. Now, those toxins are stable to heat. In other words, you can boil that for up to, I think it's 40 minutes, and it's not going to disrupt the toxin. So cooking the chicken salad at this point is not going to help. This can also happen with hamburgers. You get it in there, you cook it, it's well done, you think you're okay. If you have large amounts of toxin, it's not going to do you any good. So that's the mesophilic temperatures and time. I, the customer, go and order the hamburger or the chicken salad. I eat it. And that toxin causes an immune response. Your body says, hey, wait a minute. This is full of bacteria. It's full of toxins. Let's get rid of it. Bleh! I vomit. Now, you can also have diarrhea associated with this. But generally speaking, the 24-hour flu is violent vomiting, projectile vomiting. You feel like you're going to vomit up your toes. And you feel absolutely horrible and you lay by the toilet moaning. Now, you can tell staphylococcal food poisoning from some of the other gastrointestinal distress that we will be talking about later because of how quickly it comes. In the book it says 1 to 6 hours later. I've heard of some cases that within 20 minutes of eating, if you have large amounts of toxin, you're vomiting it up. Interestingly enough, because of this, people who have had staphylococcal food poisoning will avoid that item for the rest of their lives. So, for example, if I'm vomiting up violently chicken salad, I'm not going to touch chicken salad because I think, oh, that's what made me sick. And if it's that quick, that is what made you sick. So, staphylococcal food poisoning. Most food handling practices have been put in place to avoid this. So, you clean your hands, you don't pick your nose, you wear gloves. This is to prevent skin bacteria from getting into your food. We are especially careful with high protein foods. We refrigerate or immediately go out at high temperature, and we try not to leave our food at mesophilic temperatures for long periods of time. Now let's talk about the liver. The liver is an associated organ of the digestive tract. It produces a lot of enzymes that go into digesting your food. It also filters your blood, and it's involved in monitoring your blood sugar levels. It's a big important organ. Because of that, it gets infected with an awful lot of viruses that just love to hang out in the liver and cause inflammation. That's what hepatitis is inflammation of the liver. Now, we've got a whole suite of viruses that cause hepatitis. We've got hepatitis A, B, C, D, E, F. We may be up to G for all I know. And they're not related. Okay, they are not the same. The only thing that brings them together is the fact that they infect the liver. So we're going to talk about two groups of hepatitis. There's the foodborne group, which is hepatitis A and E, and there's the bloodborne ones, which are hepatitis B, C, and D. So just think of the ones on the end come from food, fecal oral transmission. The ones in the middle, those are transmitted by blood to blood or fluid to fluid contact. Okay, so let's talk about hepatitis A first and hepatitis C. This is the one that you hear of where you have an outbreak associated with a restaurant. Okay, it's infectious foodborne hepatitis. It's fecal oral transmission, so somebody who has hepatitis uses the bathroom, has a bowel movement, does not wash their hands. They have feces with the hepatitis in it. It gets into the food, and you eat it. Now, foodborne hepatitis viruses are extremely environmentally resistant because they're set up to survive getting into the water supply until it's drunk by the next person. Since we have good water treatment facilities, we don't see that in this country. So, I eat the fecally contaminated, and it doesn't have to be big chunks of feces either, it can be microscopic amounts. I eat the fecally contaminated food, gets into my digestive tract. From there, it gets into the liver. Okay. Foodborne hepatitis is extremely contagious because from the liver, goes back into the intestinal tract, is eliminated with feces, and it can resolve on its own, but you're sick for a long period of time. You have tenderness in the region of the liver, you have fever, and because of disruption of the functioning of the liver, you have accumulation of bile within the blood, which causes the yellowing of the eyes. Okay, so I'm sick and infectious for a long period of time. We do not have very good antimicrobials for this. So the best thing is to prevent infection by good handling techniques, wastewater treatment. We also have a vaccine for hepatitis A. Now for the bloodborne ones, hepatitis B, C, and D. So here we have a picture of hepatitis B, Hep B. Now this is the usual full virus, where you have the virus inside with the DNA. You have the envelope that comes from the host cell studded with viral antigens. The most common one is Hep B surface antigen, or HBsAg. And this is what your immune system responds to. Now, the virus gets in a hurry, and sometimes it produces little spherical particles that are studded with HBsAg, and you also get the tubular ones. Okay, with the blood-borne hepatitis, you can have all of the symptoms of hepatitis, and it gets into your fluids because it's not fecal oral. You're not getting it into your digestive tract. It's not coming out of your feces, but it is in all of your fluids. It's in your saliva, it's in seminal and vaginal secretions, it's in the blood, so it's sexually transmitted or through IV drug use or unclean needles of any sort. So because we have all of these particles floating around, okay, you're extremely infectious, but a lot of people, the disease resolves and you get better. But with Hep B, about 20% of all patients go from acute disease where they have active hepatitis to being carriers. They're still producing virus. You can still pull these particles and the Dane particles, the complete HBV, out of their blood, but they don't have the symptoms and signs of hepatitis. Well, that's when you're also infectious. With these incomplete viruses that are produced, for a long time we used these as a way of producing a vaccine. Because these are heavier, you can centrifuge these and they go to the bottom of the tube. You have these at the top. And if we inoculate you with these, your immune system sees this and develops antibodies. Well, you can see the problem with this. You might accidentally get a Dane particle and be infected with hepatitis. Okay, so we've moved away from that. What we've done, the vaccine is now a subunit vaccine. We were able to do enough research that we're able to get the genetics for the HBsAg and put it into, I believe it's yeast cells, could be E. coli. Put the gene into the other microbe that is not pathogenic, have them produce the antigen, and we inject you with that antigen and you become immune. The hepatitis B vaccine is very, very good, and it's recommended for everybody because it's just too easy to have fluid-to-fluid contact with Hep B. Unfortunately, we haven't reached the point where we have a vaccine for Hep C. Hep C is more likely to cause chronic infection, and it's more likely to lead to cirrhosis of the liver or liver cancer. This is a nice healthy liver. This isn't a very unhappy liver. As I mentioned with the peptic ulcer disease, anytime you have chronic infection, you have chronic inflammation. Chronic inflammation can lead to long-term damage, either in the form of cirrhosis here, where you have scar tissue being built up in the place of healthy tissue after damage to the liver, or you can develop cancer. Okay, so hepatitis is not to be messed with. Well, that's it for part 1. Take a break, come back, and we'll talk about diseases of the intestinal tract on down.